My name is Rob Burgess. I am the Chief Business Officer for Sino Biological, and I would like to officially welcome everyone today, especially our speaker as well as our attendees, to the first installment in what I believe is 8 installments of the Sino Biological hosted and sponsored seminar series on COVID-19 immunodiagnostics. We're very happy that you could attend, and good afternoon and good morning to you wherever you might be. I want to mention a few housekeeping items first, the first of which is that the audience is muted throughout the seminar, and this cannot be unmuted, but I would like to suggest that if you have a question for the speaker, that you simply type it into the chat box, and then I get the privilege of selecting the best questions, and I get to ask those of our speaker at the end of the seminar, and we will get a live response from our speaker. So this webinar is recorded, and everyone who's attended will receive an email in a few days, which provides a link to the recording. So without further ado, it is my honor and privilege to introduce to you our speaker today. His name is Dr. Benjamin Miller. And Dr. Miller is the founder and chief scientific officer of Adarza Biosystems. And we're going to hear some exciting information about Adarza's technology today. In addition to being the founder and chief scientific officer of Adarza, Dr. Miller has been with the University of Rochester since 1996, and he is currently Dean's Professor of Dermatology, Biochemistry, Biophysics, Biomedical Engineering, and Optics at the University of Rochester. Dr. Miller received his PhD in bioorganic chemistry from Stanford University, and he did his NIH postdoctoral fellow at Harvard University in Boston. So the title of Dr. Miller's talk today is Analyzing the Human Antibody Response to Pandemic Respiratory Viruses with Adarza's new ZIVA platform in high-definition antibody arrays. And this is a case study on COVID-19. So with that, Dr. Miller, I will turn it over to you.
Thank you, Rob, and appreciate the introduction and really appreciate the opportunity to present our work to you today. Let me get my slides up and we'll get started. So as Rob mentioned, I'm going to tell you about how we're using Arrayed Imaging Reflectometry and Adarza's ZIVA platform for analyzing the human immune response to COVID-19. And this work actually grows out of a long effort in my lab on developing new tools to analyze how humans respond to upper respiratory viruses. So just to give you a little bit of background, obviously this is the kind of image that we've all become very familiar with over the last few months. This is from Google's coronavirus information site, and if you're like me, you go there every morning to find out what's happening in your location or worldwide about what the coronavirus is doing. And so as we began to see that the coronavirus epidemic was growing and growing rapidly, it became very clear to many people, including us, that diagnostics would be incredibly important as we worked to help limit the spread of the pandemic and also develop new methods for combating the pandemic, whether that's via therapeutics, vaccines, or basic biological studies to understand how the virus works. We are focusing on antibody assays, that is, using antigens from coronaviruses and other respiratory pathogens to understand the human immune response. And the reason we can do that is because we have a technology that's able to very sensitively, specifically, and quantitatively detect those antibodies in a blood sample. That technology is Arrayed Imaging Reflectometry. This is a label-free detection system that has been developed by my lab at the University of Rochester and is being commercialized by Adarza Biosystems in the ZIVA platform. So how this works, well, to put it in a fairly simple analogy, if you're like me, you're really obsessive about your computer screen. At least I am. And inevitably, you know, I'll clean off the screen perfectly so it's beautiful. I can see everything in the screen. And someone will come into my office and they'll point something out and they'll touch the screen, leaving a nasty fingerprint or thumbprint on the screen that I then have to look at for the rest of the day. So what you're doing there is you're detecting biological material, whatever has come off that person's finger or thumb, based on the change in reflectivity on the surface of your screen. And that's really what arrayed imaging reflectometry is about. What we do is we take a silicon chip with a coating on it that we very carefully control, and we set that up so that spotted capture molecules, in this cartoon, they look like antibodies, but they can be antigens, antibodies, peptides, DNA, RNA, what have you, anything. We set that up so that light interacting with this chip completely interferes with itself within that thin film and no light comes out. Actually, we can make this, well, let's use another analogy. If you have glasses that you wear, the typical antireflective coating on eyeglasses is somewhere between 10 to the third, 10 to the fourth. So 1 in 1,000 to 10,000 photons that's incident on your glasses will reflect off. We can control these antireflective coatings on an air chip to up to 1 in 10 to the 8th. So only 1 photon in 10 to the 8th incident on the chip reflects off. So if I'm looking at this with a CCD camera, all I see is a set of dark spots on the background of that chip. And then if some molecule binds to whatever my capture molecule is at a specific location, that perturbs that antireflective condition and allows light to begin leaking out. And the amount of light that I see on a particular spot is corresponding to however much material I've captured. So it's completely quantitative, very sensitive, and then we know what's what just based on where we are in the microarray. So this can all be implemented in a 96-well plate format, and I'll show you more of that as we go along. This is a technology that, as I mentioned, we've used quite a bit for understanding responses to respiratory viruses, and in particular via the National Flu Center network, we've done a lot of work looking at influenza, either with arrays of antibodies to profile viruses, or along the lines of what I'll talk about today for coronaviruses, using antigens from the viruses to look at antibodies. So just briefly to go over a couple of these applications, here's one example where we took an array of 85 human monoclonal antibodies. Each monoclonal antibody is arrayed in 2 spots in this particular example. So these are all pairs of spots that are a single antibody. And then we exposed arrays to different flu viruses. This one has been exposed to an H7N3 virus, and this one has been exposed to an H1N1 virus. And you can see very easily, just by the naked eye, that the patterns of response for these 2 85-plex arrays are very different, okay? So we can actually use this like a crowd on a chip way to serotype different kinds of flu viruses. And that works also for closely related viruses. This is an example here where we looked at 115-plex arrays exposed to 2 different H7N9 viruses. This is Shanghai H7N9 versus Anhui H7N9. These differ only by 6 amino acids in their hemagglutinin, and we can very easily tell those apart on this array. So it's turned out to be a very interesting, useful tool for serotyping flu viruses. More recently, we used this in a nationwide mock pandemic exercise that the NIH ran last year, where they asked the entire flu research network to basically pretend that a couple of different canine flu, dog flu strains had suddenly become infectious to humans. And the goal here was for NIH to find out, well, how would the network respond? How would all of these researchers develop tools and response protocols that might be useful in the context of a viral pandemic? And so a very large group of researchers, this is the author list from the paper that came out a couple of months ago in PLOS Pathogens, went to work trying to understand these viruses. In our case, we used our 115-plex monoclonal antibody arrays to say what flu strains are most similar to these mock pandemic strains. And very quickly, we were able to show that Hong Kong 68 had the closest relationship, at least among the strains that we had in our hands, for these mock pandemic strains. Now, what that means is that if this were a real pandemic, we could use vaccines against Hong Kong 68 against these canine viruses and potentially expect that to have some protection. We can also look at the antibodies that respond very well to Hong Kong 68 as well as these pretend pandemic strains using this sort of format. And identify monoclonal antibodies that might be useful as antibody therapeutics and as ways to identify potential targets for vaccination. On the antigen array side, we've used antigen arrays made up of influenza hemagglutinin, that's this protein right here, to detect antibodies both in humans in a candidate vaccine trial for an H5N1 influenza, as well as in this particular example I'm showing, which was for viral surveillance, where in these graphs, what we're looking at are serum samples from ducks, mallard ducks that had been infected with different flu strains. And we were able to quantify specific immune responses by the ducks to, for example, H4N6 flu, that's the H4 antibody response, H6N2, also a very strong antibody response. And when you look at some of these others, this was an H3N8 virus, and you can see there's not much of a response. Likewise, H12N5, some response, but not a ton. Both of these birds really didn't get very sick. So that was consistent with what we saw in the antibody response. It's also useful in that we could do multiplex serology. This was, again, the 18-plex hemagglutinin array, looking at a series of serum samples from mallard ducks from the Cape May, New Jersey area. And this is something that's pretty much impossible to do with standard ELISA assays because there's only so much serum you can get from a bird, but we could profile all of the different antibody responses to these different strains, from individual bird samples. And it matches up quite well with standard hemagglutination or viral neutralization assays. So it was specifically these two things, one, that exercise of going through a mock pandemic, plus what we had learned from making these hemagglutinin arrays for influenza, that led us to start thinking about how we might contribute to the response to SARS-CoV-2. And in particular, since many people have been working on antibody assays, our goal was to do really three things. First, to quantify, to provide quantitative information about how much antibody was present in a sample. Second, to use the multiplex nature of this assay to be able to discriminate. It doesn't do you much good if your assay can't tell the difference between a COVID-19 response and a common cold, a circulating coronavirus response. So by having multiple different antigenic probes on an array, we can begin to tease apart those different responses in a way that's very challenging to do with just a singleplex or duplex assay. And then finally, the goal of any of the studies that we do is to enlighten. What can detailed high-resolution information about the human response to SARS-CoV-2 and other viruses tell us about COVID-19 disease? So that was the goal. So what I'm going to show you in the rest of the talk today is initially some of the preliminary validation work that was done in my academic lab at the University of Rochester Medical Center. And then I will show you how DARZA Biosystems picked up on that work and extended it to take what we had done, implement it in the ZIVA automated analysis system to provide a tool that now gives a very high throughput, high resolution information about COVID-19 and responses to other upper respiratory pathogens. Like many folks who've been developing antibody assays, we have focused on antigens from the spike protein of SARS-CoV-2 as our primary capture molecules. In particular, you'll see that we have proteins corresponding to full-length extracellular domain of the spike protein, that's S1 plus S2 domains. We also have S1, S2 alone, and then the receptor binding domain alone. That's the point of contact between the coronavirus and the human ACE2 receptor. In the initial work that I'll tell you about, we didn't have nucleocapsid protein. That's turned out to be one that is quite interesting. So that's on the DARZA version of the array, and you'll see that later on. And I'll say that for all of our development, all of the coronavirus antigens were obtained from Sino Biological. You know, they haven't asked me to say this, but I'm going to say it. Sino has been a terrific partner through this process and for many years. In fact, all of our flu antigen work back, let's see, we've started that several years ago, has been done with proteins that we've obtained from Sino Biological. So it's been a great resource for us. These are all obtained from the baculovirus expression system and their His6 tag. We don't use the His tag, but rather are using random orientation amine reactive surfaces to immobilize our proteins. Here's the initial array map, just to give you a sense of what this looks like. For the first tests, we used a 14-plex array duplicated on a single chip. So what that means is that each probe molecule, for example here, the human coronavirus HKU spike protein is printed in 6 replicate spots, okay? In this position, and then again here. And likewise, the other antigens, MERS S1, MERS RBD, SARS S1, SARS RBD, SARS coronavirus 2, S1 plus S2, SARS-CoV-2 S2, SARS-CoV-2 S1, SARS-CoV-2 RBD, then 2 more circulating human coronaviruses, that's 229E spike, OC43 spike, and then 3 of our reference hemagglutinins. This is Brisbane flu B, and then 2 flu A strains, California H1 and Wisconsin H3. So around each 6-pack of spots, we have an array of control spots. In this case, these are all anti-fluorescein, which is what we've used for all of our academic work. Since this is a label-free assay, we like to have those as a control for non-specific binding, and also just as a reference for the starting thickness of the chip. On these, we also have a blank area, which provides a further control in that we can easily see what's happening to the background of the chip. So in each one of these experiments, there are a ton of control areas that can be used. And also, as you think about the data that's coming off one of these chips, each spot is about 110 pixels. So 110 pixels times 6 spots times 2 locations, you have a lot of statistical robustness in the data that comes off a single chip. So these were all printed using standard methods. And then, of course, the first thing we wondered is, do they work? So to validate the assay initially, we used a polyclonal antibody that we also obtained from Sino. These are the data here. You can see array images. This is an array that has just been exposed to fetal bovine serum, which is our reference background matrix. You can see that most of the spots are dark. There are some of them that are a little bit bright. That is just a formulation issue in that early on, these printed a little bit thick. That's something that we've subsequently fixed, but I've left these images in there. If we expose that though to 1 microgram per mil solution of the polyclonal antibody in fetal bovine serum, you can see that now all of the SARS-CoV-2 antigens light up very brightly, or I should say all of the RBD containing antigens light up brightly because this polyclonal was raised against RBD. There's no response to the S2 protein, which is as it should be because that doesn't have the RBD within it, but all the other SARS-CoV-2 antigens light up. We get a little bit of a non-specific response on one of the SARS RBD as well as MERS RBD, only at the highest concentration, and that's not a significant problem. And as you can see, the intensity drops off as you drop the concentration. A little bit easier to see that if I extract these numbers and plot them like this, where we've fit everything to 4PL standard fit for how you would expect to see a response on an array. Everything is well behaved, quantitative. This assay has not been optimized for sensitivity, but even so, in label-free mode, we get down to a detection limit on the order of 10 nanograms per mil or so. We could certainly optimize that and extend it quite a bit farther down. But given what we know about antibody amounts in typical COVID-19 patients, this is plenty sensitive. And as you would expect, again, throughout the titration, really no signal on the S2 protein. So great, we can work with a polyclonal antibody. What about human serum? Well, initially when we did this work, there wasn't really any convalescent COVID-19 serum to be had. So we started scouring the lab and our friends and neighbors and got a set of initial samples to screen. Here's what we got from those samples. I'm only showing the coronavirus, SARS, MERS, and SARS-CoV-2 antigens here in this graph. And what you can see is that for most of the subject human samples, these are at a pretty low level. There are obviously a few outliers. Here's one sample and another that had an extremely strong RBD response. These two are pretty interesting because they have responses to several of the SARS-CoV-2 antigens and a little bit less here. But really, it wasn't clear what this meant. We can definitely go back to these subjects and say, well, you know, what's going on here? I should have said at the outset, we acquired all these samples from people that had said they hadn't had any COVID-19, hadn't had any contact with COVID-19, but going back and talking to a few of these subjects, this person and this person, both of them had significant others who had had active COVID-19 disease. So in all likelihood, both of these people actually had COVID-19, were just asymptomatic. These two, actually an interesting thing about these two people, given the similarity of their response to the different antigens, this person is this person's spouse. And although neither of them had had any contact, known contact with someone with COVID-19, they had both spent quite a bit of time in Seattle at the time of the initial outbreak there. So it's possible they came into contact and with someone that had COVID-19 and also had an asymptomatic response. But really, you know, absent a known positive, wasn't clear what the threshold was. So after a couple of weeks of working with these samples, we were finally able to get some convalescent COVID-19 serum samples via the University of Rochester Medical Center Healthy Donor Protocol. These were all subjects who had diagnosed COVID-19. They were minimum 27 days since symptom onset, a pretty broad age range, roughly 50-50 male and female. Interestingly, even though these were diagnosed COVID-19, 5 subjects did not report a positive PCR test. And you'll see why that's interesting momentarily. So we ran these on our arrays and this is what we see. So here's an array image of fetal bovine serum. Again, this is essentially all black. You can see we've improved these antigens still a little bit thick, but much better. And I should say that even like this, we have plenty of dynamic range on the assay to be able to see responses. If we throw in pooled normal human serum, this is donor serum that was acquired well before COVID-19. So it's all serum that should not have any of the SARS, MERS, or SARS-CoV-2 antibodies present, and that's exactly what you see. These are all dark, but we have strong responses to circulating coronaviruses here, here, and here, as well as to the flu antigens. And if we add in the, or I should say not add in, if we incubate a chip with one of our known positives, now we begin to see those strong responses to the SARS-CoV-2 antigens. So quantitatively, looking at all these samples, here's what we see. You can see that there's a very strong response. This is at a 1:5 dilution. That was what we ran initially for all of these samples. We subsequently looked at a 1:20 dilution in an effort to see if that might enhance our dynamic range for positive and negative samples, as well as potentially reduce nonspecific binding. But you can see very easily that most of these show a very strong response to the different SARS-CoV-2 antigens. This particular subject did not. And actually, as it turns out, this subject also never had a positive PCR assay. So I think this person probably actually did not have COVID-19. Interestingly, this person doesn't show any antibody response to SARS-CoV-2 antigens, but did have a PCR positive result. So had real COVID-19, just didn't have an antibody response. So that's something that's going to be interesting to look at as we continue to expand the number of subjects that we look at in this assay. Of course, with any new assay, one of the things that's really critical to do is correlate that to a gold standard. And so working with collaborators here at the University of Rochester Medical Center who had a gold standard ELISA assay, singleplex ELISA assay for coronavirus antibodies, we looked at their data, and here that is. This is their results from 2 of the different SARS-CoV-2 antigens, S1 plus S2 ECD and the RBD, as well as 2 of the circulating coronaviruses. I think this is particularly interesting because you can see that some of these subjects had just enormous antibody titers to the circulating coronavirus spike proteins. These are all in micrograms per mil, and they're IgG specific responses. So, up to 300 microgram per mil IgG specific response to OC43 circulating coronaviruses. Pretty impressive. But also there are several of these with very strong responses to the SARS-CoV-2 antigens. You can see that the same subjects that gave us very low signal also gave the ELISA low signal, not quite 0, but close to it. And if we plot these and fit them to 4PL fit, here's what you get. So you can see air response in angstroms versus the IgG specific response in microgram per mil. Particularly at the high end, that gives a very good correlation. I think we just don't have enough samples at the low end here to correlate well. But also RBD gives a very good correlation between our assay and the singleplex ELISA. So that's very encouraging that we get that, particularly since the air results are total antibody. So not just IgG specific as the ELISA assays are. Now, of course, we can make air results IgG specific or IgM specific by adding a second step. We did that here with a series of samples where we took 3 separate chips. One was just exposed to serum from the particular subject. Both looked at pooled normal human serum, 2 different COVID negative samples, and then 2 COVID positive samples. Another sample was exposed to serum, followed by an anti-IgG step, and then another was exposed to serum, followed by an anti-IgM step. And you can see that we can very easily pick out, and I'm gonna have to move my video window a little bit because I can't see my pointer. we can very easily pick out where the IgG and IgM-specific responses are, both to SARS-CoV-2 antigens, but also in some of these circulating coronaviruses and flu strains. So we can get a lot of information about those antibody-specific responses in this manner. So as I mentioned, as we were going through this process, DARSA developed their own version of this assay, now using the fully automated ZIVA workstation. And the rest of the data that I'll be telling you about is on the ZIVA workstation. DARSA was kind enough to get me one of their pre-release instruments, and we've been really running it through its paces here at the U of R. What's fantastic about this assay is that now we've got the same multiplex capability, but only requires a tiny amount of serum because it uses a consumable that's low volume. So for example, we're using 2.5 microliters of serum diluted 1 to 20 in this. So we're at a level that we can do finger stick blood quite easily. The system, you can see what the interface looks like here, basically takes, as I've talked about the technology earlier in the talk, we can go anywhere from tens to hundreds of biomarkers from a single sample. We can do both protein and antibody detection, low sample volume requirement. Depends a little bit exactly what this is, whether you're looking at antibodies as we are with the SARS-CoV-2 and upper respiratory infection panel, or if you're trying to do high sensitivity detection of something like cytokines in a serum sample, you might use a little bit less dilute material. And this allows you to access really large plex arrays quite easily. Workflow is essentially what I've already shown you. I'm actually going to skip over this slide in the interest of time and focus on this one, in that what you do is you add samples to your consumable that's on strips in a 96-well plate. That's not to say that you have to run a whole 96-well plate at once. You can run a single strip of 8 or however many you want. You incubate this 1 to 4 hours at room temperature or 4C overnight. For something like this SARS-CoV-2 and upper respiratory panel assay, we're doing that 1 hour on the benchtop, so very fast, simple. And then if you want to look at a serotyping Ig, you can add that in and incubate a little bit more, or you just, after your 1 hour incubation, load this on the ZIVA instrument. 96-well plate will be completely processed and the data delivered to you over the internet in anywhere from 25 to 45 minutes. So it's very quick. Hands-on time is 20 minutes or so, and you get the same high-quality data, actually better from this automated system as we're getting with the laboratory system. So DARSA is rolling out 2 different upper respiratory serology panels. One of them is a broad screen that includes SARS-CoV-2 antigen targets, including nucleocapsid, as well as others that I've been describing, and 1 mutant variant. MERS, CoV, and SARS-CoV-1 antigens. The circulating coronaviruses, 4 types, but then also RSV, and 9 different influenza types. There's also a coronavirus footprint antigen array. This is a 25-plex that includes a large number of spike mutant proteins, as well as the same coronavirus targets that I mentioned here. So I think these are going to be very useful tools for looking at human serum samples, as well as I'll show a little bit, monoclonal antibodies, either for vaccine or therapeutic development. DARSA also has a series of arrays that I won't talk about in much detail today, focused on cytokines, either as a screening array in 50-plex or ultrasensitive arrays in 15-plex, mouse arrays, very similar, and rat arrays that will be coming online a bit later. So those are certainly things that I'm happy to talk about, or refer you to the folks at DARSA to talk more about. So as Adarza was validating their assays, this is a set of data that they generated to make sure that the assay worked. First, using pools of pre-COVID-19 samples, or I should say individual donor pre-COVID-19 samples that I pooled on this graph. These are these blue bars, which I apologize are a little bit difficult to see, but you can see that, as you'd expect, the response is all quite low for SARS-CoV-2 antigens. And then if we look at COVID-19 convalescent serum samples, now you get a very similar type of response that we saw with our convalescent population. Most of these have very strong antibody responses. Again, a few don't, though, and all of these samples were PCR positive. So again, I think understanding why some people don't produce a robust antibody response to SARS-CoV-2 antigens is going to be an interesting aspect of fundamental research going forward. For us, we ran all of our same samples plus more using the ZIVA array. This is just what some of the data look like. You can see that I've put a red dotted line to show where the background is. This is the fetal bovine serum control sample. And you can see that much like we saw in the laboratory assay, we get variable responses, but most of these are quite strong to the COVID RBD protein. We also have an RBD protein from another source on this array, and it responds very similarly. I'll show you a more detailed correlation momentarily. But then also the other antigens, N, S1, S2, S1, and so on. So able to get all of these data. The difference with ZIVA is that we got these data in an hour and a half rather than taking much longer time using our laboratory manual system. So these kinds of multiplex arrays do allow you to start looking at all sorts of correlations. As I mentioned, we have two different sourced RBD proteins on this particular array. These are all correlations to the sinobiological RBD that's on the array. You can see that the other RBD correlates perfectly. I mean, that's as close to an R squared of 1 as you can get, I think. So that's, again, very encouraging, both for how the assay is performing, as well as for the quality of the proteins that we're sourcing. COVID S1, S1, S2, and other coronavirus antigens all also correlate quite well for these samples, as you would expect. And then we really don't see correlations to other coronaviruses like SARS, MERS, or most of the human coronaviruses, and obviously none to flu. But we do see some correlation possibly to the human coronavirus 229E. So that's one that we're going to be looking at in more detail as time goes on. And then also, of course, we see correlations between different types of flu on the array, again, as you would expect. So this, I think, points to a lot more fundamental research that can be done. For example, as we look at patients coming out of COVID-19, do the ones that had easier response to the disease, did they have a larger initial titer to some of the circulating coronaviruses? We'll see, but that's information that could be quite helpful, as well as things like, as we begin to look at patients over a longer term, how does their antibody repertoire change, and does it change differently for SARS-CoV-2 versus some of the other antibodies that are already present in their serum? Obviously, we can also do things like measure titers by doing dilution series. I won't go into detail on these data, but just to show you that we've done this, and it's actually quite simple to get a standard titer from the assay. We can also look at monoclonal antibodies. I'll just show you one example of that. Here again, I've put the red dotted line to show you where fetal bovine serum is. These are 9 monoclonal antibodies that were kindly supplied to us by Active Motif. You can see that these were raised against the RBD domain, and they really don't have any response to any of the other coronavirus antigens, which is a good indication of their specificity. But they give robust response for 8 of the 9 to the RBD antigen. Interestingly, there's one monoclonal that really didn't respond to RBD. I think this may have been a test for us to see if we could pick up the non-responsive monoclonal, but the others all respond quite well. So as things like the mutant array come online, I think that's going to be a really exciting way to also test these monoclonals so we can see whether they're responsive to the different possible mutants that are going to be present in the community. And we're eager to start testing those sorts of questions. So finally, I'll just sum up and then move on to questions by saying that hopefully I've shown you that AIR allows you to develop quantitative multiplex detection of antibodies to SARS-CoV-2, along with all of these other upper respiratory disease pathogens. The performance is correlating quite well to singleplex ELISA. I didn't show it in these data, but for the influenza work, we have also correlated all of those responses to reference ELISA assays. And so this is all quite robust analytically with reference techniques, but now providing the multiplex quantitative information now in a single well, a single experiment. In my academic lab, we're continuing to use the home-built version to rapidly modify these arrays. As I mentioned, we're really only limited by our imaginations in that we can incorporate mutants, other viral antigens, we can mix antibodies and antigen arrays and so on. But then we're excited to have the ZIVA system in our lab just because of its ability to give us a lot of data very quickly, very easily on the standardized array. And to that point, we have IRB approval for acquiring up to 2,000 samples from the community. We're beginning to acquire those and are trying to prioritize subjects who have had diagnosed COVID-19. That's going to form the basis of a lot of really fundamental information for us about how the disease progresses, who responds in what way, and how we might focus therapies and vaccines. So we're talking with various folks about clinical trials and vaccine development. So I think there are a lot of exciting things that we can do with this tool now that we have it. So with that, I'll just close. I'll thank the folks who did the work in my lab early on. We were closed down. So after Joe Buchkowski did some initial work and then had to vacate the lab given a family situation that he had people at home that really were potentially susceptible to COVID-19. Others in the group, particularly Dan Steiner and John Cognetti, were able to stay on and do a lot of the initial development work. They would hand data off to Joe and Ethan Luta and Alana Close to analyze. So it was really a great pipeline of work to get things done. And then we also had help from Michael Bryan early on in getting some of those early samples I mentioned. Of course, more recently, everyone has come back to lab, and so we've been able to get everybody engaged in this full time, and that's been an exciting thing to watch. I've had great collaborators as we've gone along, particularly via the NIH Flu Center. The local node of that is the New York Influenza Center of Excellence, or NICE, that's led by David Topham. And collaborators within that group, particularly Mark Sangster and Feng Wen, were instrumental in providing us with the ELISA data. Of course, I want to thank Darza Biosystems for their terrific support as we've developed this and for now turning it into a tool that I think is going to be extraordinarily useful, not just to us, but to the broader community. So thank them, thank these folks for funding. Once again, thank Sino Biological for giving us the opportunity to present these data, and thank you for listening.
Thank you, Dr. Miller. We really appreciate that excellent talk. Jen, if you could turn my video back on, I will jump right into the question and answer session. And again, we also appreciate the kudos and the nice things you said about Sino Biological, Dr. Miller. It's interesting, the very first question that we got as you started your talk was, your system being utilized to test IgM. And I think you gave a very nice example of that, right? Where you use anti-IgG and anti-IgM antibodies in your system to distinguish the two. Is that correct?
Yes. So that does require an additional step in the workflow, but all you do is add in, after that initial incubation, you add in an anti-IgM, and that provides additional signal that tells you what the IgM-specific response is.
Right. And this is a very technical question, but I thought it was a good one. And regarding the reflectometry matter that you, I guess, coat the chip with, somebody had asked, do you pre-calculate the mass of this in comparison to the mass of the antibodies or the antigen that you're going to spot? Is there a relationship there?
Yeah, so really that's an insightful question. Really what we are is a mass detector. But one of the interesting things about this technology is that in a way, it doesn't matter how big the capture molecule is. Because what we can do is we can tune that initial chip thickness and the way we deposit the antigen or antibody, if we're doing an antibody array, so that we start at a 0 point. And then whatever you're detecting is a change relative to a 0. And just like you learn in 7th grade math, if you divide anything by 0, you get a big number. So it allows us to have very sensitive detection of anything from small molecules to very large molecules, even beyond antibody size, using this method.
That's great. And somebody asked about the assay time. I think you outlined that in a flow chart. I gathered that it was several hours with about 22 minutes of hands-on time was the fastest. Could you say a little more about the assay time?
Sure. So it depends on what you're trying to detect and what sensitivity you need. For these SARS-CoV-2 antibodies, and really for any of the antibody assays that we run, for any of the upper respiratory pathogens, the titer of the antibody is typically pretty high. And so it's easy to detect using our system, even with fairly significant dilution. So the total incubation time, right now we're using an hour. It could probably go less than that. But it's about, however long it takes you to load a plate, which for us is around 20 minutes. And then you just let it sit an hour and then stick it in the machine.
Sounds great. And the sensitivity, did I catch that? It was at the lowest level is about 10 nanograms per mil, is that right?
So again, we have not optimized these assays for sensitivity. So with the laboratory tool, we're seeing on the order of 10 nanograms per mil for the antibody. For other targets that we're looking at, for example, the cytokine array is on the order of, for the screening array, that's on the order of a few picograms per mil. And for the ultra-sensitive version, that's now in the femtogram per mil regime.
Great, great. And we did notice that there was a light signal in your FBS panel. Can you comment on why you might have seen that?
So with any serum, you're going to get a little bit of non-specific binding. So you do see a little bit of a signal. That's why we have all of the different controls arrayed on chip, whether it's a non-reactive protein that we're just looking for a non-specific adhesion of material to the chip, or a little bit of adhesion of material to the background. It's actually surprisingly small though, and does not contribute significantly to the signal that we observe.
It's pretty much background then.
Yeah.
Okay. Yeah. And another good question by a researcher, do different types of samples, I guess the matrix that they're made out of or whatever, does that affect the success or failure or reproducibility of your system?
Not really. I'll say that we've focused mostly on serum, but we have done some work with saliva, not in the upper respiratory pathogen array, but in a different application. And saliva we've had good success with. there are all sorts of other biofluids that one might want to look at from humans and other sources. I'll say that we've tried some agricultural applications and have had good success with those. It does sometimes require optimization of the washing steps, but so far we've haven't had any problem doing that.
Okay, great, great. Question I'm reading off the chat box here. Is there an explanation as to why the receptor binding domain reactivity is superior to what you observe with the S1, S2, or the full length in your setting? I'm not sure if that's an accurate question.
No, that's a good question. Again, really insightful observation. So what a lot of people see is they get stronger response to S1 plus S2 or the full length spike versus RBD. And of course, the receptor binding domain is contained within S1 plus S2. One thing that's different about our assay is that you're looking at binding to a particular amount of protein per unit area. So if you think about the size of RBD, small protein, you're going to get a lot of RBDs down on a spot. But S1 plus S2 ECD, much bigger protein. I'm using my hands like it's a basketball. Obviously, it's different shape, but you don't get quite as many molecules per spot as you do for RBD alone. So the perceived response from S1 plus S2 is smaller than it is from RBD. That's why it's important for us to do these correlations to ELISA assays or other reference assays. But once we have those correlations, once we have effectively a calibration curve, then that difference in density really doesn't matter.
Great, great. And this is a technical question, maybe a trade secret, but I'll ask it anyway. Does the laser from the air technology cross the liquid meniscus in the microplate?
So I should have made that clear. Actually, that's not a trade secret. It's something we published. For this implementation of air, these assays are run dry. So what we do at the end of the assay is we basically rinse the chip, dry it. So there's no aqueous interface at all. We have published an aqueous version of the technique that actually allows you to do kinetic measurements in an array format, but that's a little bit trickier to implement, as you might guess, and really for these kinds of applications doesn't provide you with, certainly for research applications, that's great to have, but for what we want to know here, just concentration of molecules in a sample, it's not needed. So we focus on the dried technology.
Great, great. A little more of a business related question here. How much is the system, I guess, list price?
So I am a science guy, so I'm not the best person to answer that. But what I'll say is that both the instrument and the consumable are competitive with other assay systems on the market. And certainly if that's something that you would like to explore further, I'd encourage you to contact folks at Adarza, and they'd be happy to give you more information.
Great. And then related to that, and maybe you know that the Adarza system's been around for a while, so do you know how many units are in the field now? And can you mention maybe some like FDA approved diagnostics that are being run on the system?
So actually, so although the technology has been in development for a while, the ZIVA product is not actually in release yet. So it's brand new. I just got actually the first system outside of Adarza proper got delivered to my lab roughly a month ago. So I'm customer number one, I think. And so this is something that will be rolling out to customers, early adopters in the next month or two, and then the full official launch is later this year. The focus is really on research use. Although this is a system that we think could move in the direction of FDA-approved diagnostics, that's not the business focus of the company early on. So we'll really focus on that research application space.
Great. Well, thank you, Dr. Miller. We really appreciate your time today. You gave an excellent seminar on some wonderful technology. I'm really glad to hear that this system is being implemented in the research community, and I hope that a number of our participants today will consider the system as it applies to their own research and diagnostics. So once again, I want to thank you for your time today, and I want to thank all of the other participants today, and we really appreciate your attendance. And with that, I believe we will sign off, and I do want to remind everybody that we have another seminar coming up this coming Thursday at the same time. So thank you very much for everyone's time today, and enjoy the rest of your day.
Thank you.